Welcome, and I'd like to introduce you to Dr. Sarah Herbert tonight, an ecologist with over 15 years of experience working on Aotearoa, New Zealand's unique biodiversity. Her research focuses on how ecosystems respond to change from invasive species to conservation efforts, often blending fieldwork with statistical modelling. Sarah has worked with a wide range of species, including frogs, reptiles, fungi, and plants. She also leads a long-term monitoring project on Hochstetter's frogs on Aotea Great Barrier Island. We're very excited to be talking with her this evening. Over to you, Sarah.
Oh, thank you so much for the lovely introduction, Michelle. Shall we crack into it? So I will just share my screen and we'll put this in presenter mode. All right, so Michelle, just checking in. Can you see that screen okay?
Yes, that's great.
Lovely. Okay, so our talk tonight has a very Jane Austen-esque theme, predators and protection. Can we help Hochstetter's frogs to thrive? But before we start going into our question, I just want to give you a little bit of background about the endemic frogs of Aotearoa, New Zealand, because I realise that the people tuning in might be from all walks of life. So we often say that the endemic frogs of Aotearoa are Leiopelmatid frogs, and what that means is that they're all in a single family called the Leiopelma. Now, this very, very, very confusing family tree on the left of the screen is a wonderful resource available for AmphibiaWeb, and that actually shows you every single family of frogs in the entire world. It's a lot to take in because, as you can see, amphibian biodiversity is immense. But what you really need to know is that in the purple, we have the caecilians. These are these really funny little sort of worm-like amphibians that you may have seen on David Attenborough's Life in Cold Blood, mostly restricted to the neotropics. We also have the salamanders, which if anyone's been living in England or Europe, your common garden newt your common newt that tends to live in ponds, that's part of this group. You see, these are two tiny little branches of the entire amphibian family. And then we have the frogs. These are the hopping little fellas that we're all pretty familiar with. And in case you're wondering, when I say the Leiopelmatids are a basal species, that means they are really, really, really distinctive. Out of all of these frogs, you can see this is the first branch that came away from the salamanders. And right here at the bottom, you can barely see it here, so I've magnified it, the Leiopelmatidae. Now, the Leiopelmatidae family are incredibly special because they are endemic to Aotearoa, New Zealand. And when I say endemic, what that means is they are not found anywhere else, only New Zealand. Interestingly enough, the closest relatives are called the Escaphidae. So this family is from the New World, and they are called tailed frogs. And what's these two families have in common is that both have retained tail wagging muscles. Yes, that's right. Because of course they split out from the salamanders, which have tails, to the anurans, which are the frogs, which lost their tails, but because they're so close to that split, they've actually retained these little tail wagging muscles, and in the Escaphidae, they actually do have a little tail that they can wag. So we hear about them a lot, and the conservation of Leiopelmatid frogs, I mean, they're found nowhere else in the world. So anything that we can do here for them in New Zealand actually means a lot in terms of conserving amphibian biodiversity worldwide. And the situation in New Zealand is that we currently have 3 extant species of endemic frogs. Those of you who are more familiar with kind of the history of our endemic frogs in New Zealand and their taxonomy might be looking at this and going, what do you mean there's 3? Didn't there used to be 4? couldn't there be as many as 13? Well, you're both right, actually. And I'll go into this in a moment. But I'm giving you basically the most up-to-date view of where we're at with the different frog species that we have. So on the left, we have this lovely little chonky frog called Hamilton's frog or Leiopalma hamiltoni. Now, these in the very latest threat classification listings, which is hot off the press this year, unfortunately, their threat status has been increased to being threatened, nationally critical. And what that means is this is the highest threat classification you can have in New Zealand. So these guys, unfortunately, are in a bit of trouble. But the good thing about, I guess, the good thing, if you can say there's a good thing about being really seriously in trouble, is that all of the populations of Hamilton's frog are in either on pest-free offshore islands or in a pest-free spot in the mainland. So there is probably a population crash happening, and Doc is working really, really hard to try to figure out why that is. Now we're actually going to move all the way onto the right of the screen and introduce a somewhat happier picture. We'll just move that box so you can see the beautiful photo taken by my colleague Carrie Knox. These are Archie's frogs. So Archie's frogs are still found on the mainland in a very few locations, generally in the Waikato area. Well, actually, sorry, all in the Waikato area, I should say. Happily, their threat classification's actually gotten a little bit better because there's been a massive amount of conservation effort put into this species, including some really intensive predator control programs. So thankfully, this one's been lifted out of the threatened classification and is now considered at risk declining. So we're not in the clear, but it's a happier picture than before. Now to move on to Hochstetter's frogs. This is Leiopalma hochstetteri. This is where it gets quite confusing. So when we speak at the moment, and the way I will speak about them is as Hochstetter's frogs, but we know that there's evolutionarily significant units of the species, and what that means is that there's quite a lot of different populations, some of which are separated geographically, but there's actually enough genetic diversity within these populations to consider them as possibly different species. But if not different species, definitely genetically distinct and probably on their own evolutionary pathways. So it makes really good sense to try to apply conservation to all 11 lineages, even though at the moment we're still not quite sure whether they are different species or just very distinct forms of the same species. And accordingly, their threat classifications are, it varies. They go all the way from being at risk declining to threatened nationally critical. That's one population that it looks like in the latest evidence, their streams have become really trashed by a bunch of cyclones that we've had hit the country, particularly the East Coast recently. Yeah, so it's complicated, but I'll explain more. Before I move on from the slide, though, I should probably add that there are fossil records that suggest that we have at least, well, I should say we know that we have at least 3 extinct Leiopalma species as well, the Aurora frog, Markham's frog, and Waitomo frog. There was a lovely guy called Luke Easton who did his PhD thesis on the genetics and on like the fossil record of what we know about frogs. I don't think he found any more, and I think that's why we've settled on just these 3 species for the moment. But who knows, as we find more fossil deposits in different caves around New Zealand, the number might grow. Now, because it's these different evolutionary significant units are a lot to get your head around, so I thought what I would do is just kind of show you where Hochstetter's frogs used to be. And I mean, of course, Hochstetter's frogs is just the whole unit. So there's actually fairly good evidence that they used to actually be spread all the way down to the west coast of the South Island, just up on the top here. And they probably also used to be found right through the upper half of the North Island, with the exception of the really northern reaches of Northland. And basically what I've just done here is I've just gone through the latest threat classification listings and tried to pick out where each of these different evolutionary significant units are found. If there's any experts listening, apologies if I've gotten the Ottawa population wrong, but I think from what I've been reading, I think it's around here in the Bay of Plenty. So the main things I think to take away from this figure is that we're looking at a species that has had quite a big range contraction over time. And what is I'm going to speculate a little bit here, but what is really interesting sometimes is when you see these big range contractions, what historically would have been just a decline in genetic variation can become arbitrarily separated. And then, of course, that kind of sets each population off on its own evolutionary trajectory. Again, totally speculating here, but we have seen this in some other species of lizards that have had a similar type of historical fragmentation, both in New Zealand and Mauritius. So it's just an interesting little thing to keep in mind. But nevertheless, we have a lot of genetic variation, and we need to be giving conservation effort to all 11 of these units separately. So how do we actually do that? Well, I think it's a really good thing to just introduce some of the key threats. And the things that we know are a problem right now are non-native predation by non-native species and habitat loss. I think of interest to listeners who are doing work in the Waitakere Ranges is habitat loss does seem to be an ongoing issue in the Waitakere Ranges, because I've heard from consultants who work around the Auckland area that there's a bit of an encroaching problem where they're getting new subdivisions starting to encroach on the boundaries of the forested area in the Waitakere Ranges. So basically, these frogs really need a forested canopy and a clean stream to thrive. And if they don't have these two things, then we expect that the numbers will go down. However, do feel free there's a few caveats in that, and people have found a few exceptions. So feel free to ask me about that in more detail in the questions. Um, Climate change is always a big one when you're talking about cold-blooded animals or ectotherms. So there is an excellent paper by Germano et al. That was published in 2023, and they did basically some modeling of the species distribution, and they looked at how different climate change scenarios might affect Hochstetter's frogs and Archie's frogs. And what they what they are predicting from their research is that the climatic envelope of Hochstetter's frogs is going to move further south into cooler regions as climate change progresses. However, unlike Archie's frogs, which is kind of a nice story of potentially having an expanding range, it seems that the range of Hochstetter's frogs, the top regions are going to get too hot for them, and they're probably going to need to be moved south, which is a really challenging problem when we've got all of these very genetically distinct populations that are actually quite restricted to different areas of New Zealand. So that is an issue that we are all going to have to think about on the horizon. Happily though, so amphibian chytrid fungus, otherwise known as Batrachochytrium dendrobatidis, which is BD because the former is such a mouthful. This fungus was introduced into New Zealand, and we think around the mid-90s, and there was quite a big flurry of work actually to try to figure out how threatened our native frogs would be by this fungus. Um, at the moment, it doesn't seem to be too much of an issue. Hochstetter's frogs they don't really have a very high susceptibility. In fact, they're very hard to infect with BD. And there's actually not been any recorded cases of infection with BD on Hochstetter's frogs in the wild. So keep our fingers crossed that that's one thing we don't have to worry about for the present. Now, because this is a talk that is aimed at a predator-free group, I thought it'd be really interesting just to do a quick overview of the evidence about, well, which predators actually are a problem. We know that predation is a problem. And I think the key thing that really sticks out here is that there's actually still quite a few unknowns. So we do have, I think, really good evidence that ship rats and stoats are a problem for Hochstetter's frogs. And that's because both of those species are non-predators. So that's, either predation has been seen or they've been found in stomach contents or basically in or their DNA has been found in poop from these species. So again, we know they eat them. And furthermore, I think what's really, really compelling was a study done in the Hinua Ranges that was also published in 2023. That's by Michael Crosland and his colleagues. They've actually, I think they've got really good evidence that both ship rat and stoat control programs aimed at kōkako actually benefit Hochstetter's frogs as well. And that study was done using, for those in the know, I think, using really robust methodology too. But the rest is all a little unclear. So we know from, I'm going to keep citing her work a lot, but we know from another study by Germano et al. In 2023 that mice do affect Aotea's frogs, that we don't really know whether or not Hochstetter's frogs are affected too, but just something to keep in mind. Possums, interestingly, don't actually appear to eat either native or introduced frogs. I could only find one record of a ferret eating a frog, and that was an introduced frog. Pigs, we know, will predate on introduced frogs, but Hochstetter's frogs have, this study was actually really cool because it was done in the Waitakere Ranges. Hochstetter's frogs were never found in pig stomach contents, but we do actually think that they trample and they root a lot around the stream edges, and we think they probably disturb stream integrity. And that's actually something that we see a lot at our study sites. And then with cats, again, we know they predate on introduced frogs, but we don't know what effect they have on Hochstetter's frogs. So I think the main thing is I think here, if you have a predator control program that can get on top of stoat numbers and on top of ship rat numbers, then that's a really, really good start. But I still think it's really worth keeping in mind that there are a lot of other predators in these systems and they may or may not have an impact. So I think until we have more evidence, it's probably worth trying to control them all, except for possums. But of course, possums nobble the canopy, so gotta control. You just gotta control them all. And so that brings us to our work. So I've been really, really fortunate to work with a lot of amazing people, including iwi's, Ngāti Rehua, and the Windy Hill Sanctuary Trust, and several other wonderful, wonderful field assistants and experts. I've been working with Hochstetter's frogs up in Te Paparahi, which is on the northern part of Great Barrier Island, for 13 years now. And the initial aim of our program was actually to just see what the numbers were like up there, because at the time, Windy Hill Sanctuary was quite interested. I should say Windy Hill Sanctuary, which is down here at the bottom of the island, was quite interested in seeing whether it would be feasible to translocate Hochstetter's frogs from the northern part of the island, which this is where most of them live, down to the south. So in 2012, we started by going up there, surveying 15 transects, which you can see are all in forested and often very, very steep gullies, for good reason. Interestingly enough, we actually find the best numbers in the steepest gullies, which is great for the frogs, not so great for the surveyors. So it's incredibly challenging work. But to tie it back to the ESUs, the way that this actually ties in nationally is this is the only study that's currently being done on the Great Barrier Island ESU. So, well, we think quite important stuff, but possibly biased. So basically what our work involves is we've gone back to these streams 4 times now since 2012. So that was 2012, 2015, 2021, and very, very recently in April 2025. We revisit our stream transects. Each of these are 100 meters long. Turns out we chose 100 meters at first because we just wanted to make sure we would actually get enough frogs in each transect. Turns out we sometimes get hundreds of frogs in some of the transects, and so now we just think we're punishing ourselves by continuing to do ones of such length. But we basically go down. This is a piece of transect tape. We roll out the tape right down the midline of the stream, as you can see there. And for the first time, we piloted a double surveyor technique. And the reasoning for that is because at the time there had been some very good repeated surveyor work that had been done using occupancy modelling. But occupancy modelling, it tells you the probability that a transect is occupied. And we were very, very interested in getting really good information on how many frogs were in each of these transects. So what we basically do is we lay out the transect, we get 2 and now 3 people, because we've expanded the program. We get each person to survey their part of the transect, and then everyone swaps over, and then they swap over again. So nowadays every part of the transect is independently surveyed by 3 people. And that is quite cool because that allows us to get an understanding of what is if we see a frog, or I should say, if we don't see a frog, what is the chance that we didn't detect it? Which is quite important because that then allows us to actually estimate abundance. And it means that our estimates are a bit more robust to things like, some surveyors are better at finding frogs, some weather conditions are a bit better than others, things like that. That kind of concern you if you're a real wildlife biology nerd. But we figured at least if we were having 2 people survey, we'd hopefully be able to even just take the average of the count data and just get a bit more robustness that way. When I said the streams were steep, they are very steep. This is actually one of our best streams that you can see. It's got some big, huge waterfalls in there that are probably about 4 times the size of a person. So it's one of those things where we have to go really slowly and really carefully, but it is a wonderful workout. And again, I've just put some photos here to show you what it looks like in the field. This is from us this last year. I think we're trying to lay the transect here, and it does actually take 2 or 3 people to do this because you've got one person at the top end yelling at the person that's got the bottom end and telling them how far they've gone down and how far they still need to go. And then quite often we have a person in the middle who will be just passing that message down the chain. And I think things to note about this is actually what good Hochstetter's frog habitat looks like. And the reason we know this is because it's in a steep-sided gully. It has a really good amount of canopy, it's got quite a closed canopy, so lots of shade. They do really like cool and moist environments, and having that canopy helps preserve that. Another way you can actually tell it's got a really nice shaded environment is this profusion of mosses that are growing on all the rocks. So that's actually kind of something that we look for now when we're trying to judge habitat quality. And lastly, lots and lots and lots of rocky refugia, as well as other things like Nico fronds and logs. These are all things that are really important for Hochstetter's frogs to hide under. And indeed, while I was searching through the literature to study for this talk, I actually did find other people have found evidence that you do tend to get more babies in sites that have got a high amount of refugia, and we think it's just because the babies don't move as much, whereas the adults, even though their kind of core habitat and certainly their breeding habitat is in their streams, they will actually be moving over the ridges and into adjacent streams all the time. So even though they might look like they're restricted to streams, there's actually Hochstetter's frogs moving all around the landscapes between streams in what we call a metapopulation. And what that is basically a connected series of populations that are separated by space. Now, once we've laid the transect, we have to look for frogs. And here's a photo with a frog in it. So they're quite hard to spot, but there it is right there. this is just a tiny little, what we call neonate, and that means sort of like young of the year. It's a tiny little neonate. And in case you ever thought Hochstetter's frogs were really boring, well, while the majority of them are brown, up in Te Paparahi we get a lot of colour variation actually. So we get a lot of really green little frogs like this, greens, browns, tans, oranges, sometimes even really, really dark brown as well. So it's a wonderful population to study. And we end up with a lot of data. This is how it all gets collected. And what we do, what we've actually been like experimenting with from 2021 onwards, and that was when we got a frog expert called Doug Armstrong. And then Doug and I co-supervised a lovely MSc student called Claire Johnson. So what Claire was able to do, we're basically able to test lots of different statistical methods, and we kind of came up with this pseudo mark recapture protocol in which the frogs are really sedentary, and so if you've got 3 people searching in a row, generally speaking, if they're finding the same frog, it'll be the same frog at the same size in the same position, which is kind of cool because we can exploit that and use the frog position to kind of give them an ID. So that's been really cool. But you can see, because we've got an abundance of data being collected, I'm actually still entering it from this year, so I can't give you a lot of results, unfortunately, from this year's surveys, but we will have them ready in a few months' time, I think, once we've done all the modelling as well. But what I can tell you so far is from the paper that Claire published in 2024. And what was kind of cool, so she was looking at what's happened to the population between 2012 and 2021, because this isn't a site that has a lot of rats. When we put out rat tracking tunnels in our transects, we found about 70% tracking tunnel rates as an average. So it's pretty ratty, as is the rest of Great Barrier Island, because they don't have any mustelids, they don't have any possums. They do have cats, but as a result, there's only really the cats that are eating the rats. So the rat numbers are just really high there if they're uncontrolled. And what we found here was kind of inconclusive, as you would expect for just having 3 surveys. And I think this is where doing the 4th survey this year is going to start telling us something really interesting. Basically, all we were able to say is that the change in abundance over these 9 years, it sits somewhere between being a 24% decrease and a 10% increase. The mean was a 9% decrease. So because we've kind of got that most probable value does suggest maybe a decrease, but again, we can't really be sure because of the uncertainty around the estimate. What that's meant is that these frogs have been reassigned in the threatened category, just as kind of a precautionary measure, and we're hoping that the data from this year's work will help us to kind of fine tune and get a better picture of how they're actually doing over time. We're kind of bummed actually to find that, because when we were just looking at the 2012 data and the 2015 data, we actually didn't really see any evidence of a change in abundance, which made us think, oh, that's really interesting. They seem to be kind of doing okay when there's a lot of rats, and certainly there's a lot of frogs up there. So watch this space. But one thing that was really evident from this year's work, so we revisited after Cyclone Gabriel, and where the locals have told us that there's been a series of other pretty severe cyclones that have hit the island, and we saw a lot of this. This was formerly a really good stream for Hochstetter's frogs, and it was pretty. You can't quite see the tree fall, but a lot of these streams had a lot of tree fall in it. You can see where the banks come down. So where there used to be a lot of really good stony frog habitat is now really muddy, really silted. When the silt gets into those spaces below the rocks, that just means less places to hide. And also it tends to silt up the water, and we know that they don't like that. So when we were kind of collecting the data, we were like, oh, kind of looks like numbers are down, but again, hard to tell. And we will hopefully know in a few months. But that really brings me to the end of the story. So I just want to thank you all for listening, and hope this hasn't been too much of a whirlwind tour of many things froggy. And I would just like to invite some questions.
Thank you so much, Sarah. We will we'll be collecting those and forwarding them to you.
That sounds really great.
Thank you.